So this is the meiosis slideshow by Jackson and Jensen. So meiosis is the cell division to produce gametes for sexual reproduction. At the end of meiosis, 4 non-identical haploid gametes or sex cells are produced. It is important to note that haploid cells contain only one full set of 23 chromosomes, while diploid cells have 2 full sets of 46. The process either occurs in the ovaries or testes, depending on if the organism is male or female. There are 2 stages of meiosis. Before dividing, a cell enters meiosis, it undergoes a period of growth called interphase. In interphase, there are 3 phases which are called G1, S, and G2. In G1, the cell increases in mass. In S, the DNA is synthesized, and in G2, the cell synthesizes proteins and continues to grow in size. The next phase is called prophase, and in this phase, chromosomes become visible and the synapsis occurs, which is the pairing process of 2 homologous chromosomes, as well as the bivalent. Crossing over occurs to promote genetic variation, and the nucleus disappears, as well as the meiotic spindle forming. After prophase, metaphase occurs, which pairs of chromosomes become arranged on the metaphase plate and are attached to the now fully formed meiotic spindle. Spindle fibers from one pole of the cell attach to one chromosome of each pair, and the spindle fibers from the opposite pole attach to the homologous chromosome. After metaphase, anaphase is the next phase, which begins when the 2 chromosomes of each bivalent separate and start moving toward opposite poles of the cell as a result of the action of the spindle. Notice that in anaphase 1, the sister chromatids remain attached to their centromeres and move together toward the poles. After this, telophase occurs. At each pole during this stage, there is a complete haploid set of chromosomes. A cleavage furrow appears, and by the end of this stage, the parent cell has divided into 2 daughter cells. The separation of the cytoplasm is called cytokinesis, and the cell has divided across the equator. In prophase 2, which occurs after telophase 1, the nuclear envelope breaks down and the spindle apparatus forms. The chromosomes condense and become visible. After prophase 2 is metaphase 2. Each of the daughter cells complete the formation of a spindle apparatus. Single chromosomes align on the metaphase plate, much as the chromosomes do in mitosis. This is in contrast to metaphase 1, in which the homologous pairs of chromosomes align on the metaphase plate. Microtubules attach to chromosomes at the center. After this, anaphase 2 occurs. The centromeres separate and the 2 chromatids of each chromosome move to opposite poles on the spindle. The separated chromatids are now called chromosomes. Finally, telophase 2 occurs, and in this phase, a nuclear envelope forms around each set of chromosomes, and cytokinesis occurs, producing 4 daughter cells, each with a haploid set of chromosomes.